So I'd like to welcome you all to this webinar on male infertility. My name's Virginia Bolton and I'm co-chairing this session with Giovanni. We can show the next slide to introduce ourselves. So I'm representing the International Federation of Fertility Societies. I'm a former clinical embryologist from the UK where I worked at Guy's Hospital in London. And Giovanni, would you like to introduce yourself?
Yeah, thank you, Virginia. I'm Giovanni Corona. I'm the current secretary of the International Society of Andrology. So it's the pleasure hosting with you and sharing with you this fantastic webinar, Virginia.
So if we show the next slide, we have two speakers today, both of whom are experts in the field of male infertility. And the whole series of 6 webinars includes numerous speakers who we have great pleasure who have agreed to participate. And each of you who participate as part of the audience, you will receive certificates to confirm your attendance. And as you all know, these webinars are free to watch on the IFFS website. Next slide. Over to you, Giovanni.
Yeah, so I think first we can introduce the first speaker. That is maybe next slide. So the first speaker is a well-known authority of the field. Frank Tuttelmann is the chair of Genetic Institute in Münster, is one of the worldwide most famous author related to the genetic of male infertility. And obviously Frank will give a talk on genetic testing and counseling. So please, Frank, the web is yours.
Yeah, many thanks for the invitation and also the kind introduction. I share my slides. And it's a great pleasure to present here. Under this broad title, Present and Future of Male Reproduction. And of course, yes, I'm a medical geneticist and have been researching the underpinnings of male infertility for many years. So it's a great pleasure to be here. I have nothing to disclose, so we can start. And as I'm the first speaker today, I know that very likely everyone attending knows these infertility facts, but I always use them also when I talk to geneticists, other genetic audiences, to make clear that this is really a very common disease. Very likely more common than other diseases we always call common diseases. And what is still, maybe not in this audience, but still a bit neglected, is that in 50% of couples, a male factor is either a co-factor or the main factor. And this, I think, we altogether cannot advocate enough to make people aware. And that brings me to the diagnostics in infertile men. And the first few slides are, of course, just to setting the stage. And I put here the years when they first were used. And this is, of course, the hallmark of infertility in men is the semen analysis. And this has been around for many decades. And since the 1960s, we routinely look at the chromosomes, so through the microscope, and sometimes we find an additional X chromosome, and that constitutes then Klinefelter syndrome. Or since the 1990s, we're looking specifically at the Y chromosome for these azoospermia factor or AZF deletions. And notably, these two tests are basically the only genetic tests that are advocated in all guidelines and also applied routinely in many places. But that is, let's say, as you can see from the years, have been around for decades. And this leads to the situation that we only identify a genetic cause, as you can see here, in about 4% of infertile men in infertile couples. And this is data here from Münster. And sometimes we find other reasons like chemo or radiotherapy, but in about 70% of men, we do not find the underlying pathomechanism. And that leads to the unacceptable situation that we really have problems in tailoring the treatment, and we cannot provide personalized medicine if we do not know what really the cause for the couple's infertility is. And also, I would just like to remind everyone that we put treatment burden on the women, or if we do not understand what is going on in the men, then we go for ICSI typically. So just very briefly also this, because I need it through the rest of my talk. It's just, of course, then the standard of care in infertile men is looking at the semen analysis, and we come up with all these, and many would say diagnoses. So if we diagnose azoospermia, then of course it is clear why a couple doesn't conceive. But as a geneticist and andrologist, I would say that that is not a diagnosis. It's only a phenotype. So we see there are no sperm, but we do not know why. And this is the data from the Center of Reproductive Medicine and Andrology here in Münster. Over many decades, the patients have been collected, and there are two things to note here. One is a specific setting of Münster. This is because they are, Sabine Kliesch and her team in the surgical andrology are well known for testicular biopsies. We have a very large proportion of azoospermic men, and this is kind of special, I would say. But the other thing is not special in typical IVF centers, for example, and that is that actually 1/3 of men in infertile couples have normozoospermia, so everything looks perfectly normal. And I'll come in a few moments to an example where we learned that also in these men, some are affected by genetic mutations, and this is why they cannot father a child naturally. So what I would like to convey here and setting the stage for the remainder of my talk is that semen analysis yields actually only, if at all, we would call them diagnoses, they are only descriptive. So I would say as a geneticist, phenotypes. And we do not really know what the molecular pattern mechanism is. And that is something we and many others have been working on. And I would say quite successfully over the last years. So let me introduce you to how we changed our setup here in Münster, and that was 7 years ago. So in 2017, we were successful in getting a large collaborative funding, and this is called CRU. It's called Clinical Research Unit Male Germ Cells. And this has been going on since 2017, extended in 2020, is still running. And we, from my institution, we provide to this consortium the Male Reproductive Genomics, or MERGE, study. And what we have done since 2017, in the beginning on azoospermic men, we still do of course, the full clinical analysis, and men who underwent chemotherapy, for instance, or have other diseases, they are excluded, and then all the others undergo a karyotype analysis and AZF screening. But then we forwarded them in the beginning on a research basis, and then translationally more and more on the diagnostic level, we offer them exome on genome sequencing. So basically sequencing all the 20,000 coding genes. And since 2 years, we are now moving to genome sequencing. And actually over the years, we have started to analyze a much broader spectrum, meaning also from the other phenotypes and even normozoospermic men. And I'll now, in the following slides, in the remainder of my talk, I go through this around this circle, and for each of these phenotypes, I highlight some examples of what we have found, we and others have found out over the last years. And we have now sequenced almost 3,000 infertile men, and as this is the major focus of my research, most of them are crypto or azoospermic, so have really no chance to father a child naturally and require a testicular biopsy. So, but I actually start with normozoospermia, and here, as life plays, just because we got in touch with one specific family, we learned very interesting things. So that there are some men who the sperm everything looks perfectly normal, but they do not father a child, and fertilization doesn't work, even not if you do IVF. Only ICSI will be successful, and that is because this peculiar Caspar ion channel on sperm that is only present on sperm does not work. And if it doesn't work, the sperm do swim normally, but if they need to penetrate the zona, they need to swim more powerful, and these sperm without Caspar channel cannot do. And this is why these sperm will never fertilize an egg. And colleagues from Münster, Timo Strünker and his team, they have developed actually a Caspar test, they call it. A very easy biochemical test, and they've actually made that into a spin-off company and into an in vitro diagnostic. And so this is available. And we learned only by applying this test to more than 2,000 men who consecutively attended our clinic that actually 2% of these infertile men have cat spur-related infertility, and then insemination or IVF will be doomed to fail and only ICSI will be successful. So if we do this either biochemical test or a genetic test to confirm it, then we can really tailor the treatment for these men, and we can also prevent therapies that will not be successful. So I think a very nice example of how to translate research into the clinic and how to provide precision medicine. So another example is, and just one case report here, is this man who attended relatively young with infertility, and he has completely immotile sperm. So all of his sperm do not move at all. And he was treated since his childhood for asthma. But actually, because we then did a genetic test, we found out that he doesn't have asthma. He has a primary ciliary dyskinesia, so a multi-organ disease affecting all the cilia and also the sperm flagella, and this is why the sperm are not moving. And he requires actually a different therapy for his severe lung disease. And well, we cannot make the sperm moving, but we can perform ICSI, and then he fathered a child. And actually, throughout the following slides, there will be some gene symbols, so like here DNAAF3, but these parts are not very important. It's not about the single genes, it's about the concept I'm trying to convey. So here, I think an andrologist or an IVF center can really help for the right treatment for such a man. And then moving a bit further down this pie chart here, we are now with I call them ugly, a bit provocative maybe, ugly sperm, and also headless sperm, so acephalic sperm. And also here we learned over the last years, I mean, this is a success story, I would say, in andrology, reproductive medicine, and also genetics, because over only a very few years we have described, not we, actually many other groups have described 40, 50 novel genetic causes for such phenotypes, and these can now be tested. And this has clinical relevance because in some of these men, ICSI will be successful, while in others it might not. And this depends you cannot decide that by looking at the sperm, but you can only decide it by doing the genetic test. And also these men require activation of the oocyte to make it work. So that brings me then to our main research topic, and that is then crypto or azoospermia, so very few or no sperm in the semen, and the only chance for fatherhood is then performing a testicular biopsy and then doing ICSI if we find sperm. And of course, like with the AZF deletions, it would be much better if we know before doing the biopsy whether there is a chance to find sperm or not. And I will provide now a few examples, like highlights, showing where we are and what that helps in the treatment. So one of my favorite genes is M1AP. Margot Wirwol, a very talented clinician scientist, she found this gene in our cohort a few years ago. And then just by sheer luck, when presenting that at a conference, we got in touch with colleagues from Turkey, and we found they had this family. And both together, and we did some in vitro functional studies, showed that M1AP is causing meiotic arrest and azoospermia. And when we immediately applied that into the diagnostic setting, and so we picked up more of these men, and then we learned that actually some of them do have a few sperm. We do not very well understand why and how this works, but we now know that the success rate for testicular biopsy is relatively low, with around 10 to 15%, but of note, I mean, these are not so many patients yet described. And then, of course, the question is, okay, if this is a protein that is relevant for meiosis, so separating the chromosomes, will these sperm that look like sperm, will they be helpful for ICSI? And yes, I can tell you they can be, because just basically by a natural experiment in our IVF center, without us knowing yet that a man was affected, he fathered a healthy boy. So that is good news, but we still need further investigations to be on the safe side. Sticking one more slide with meiosis and the diagnostic year. So this is Corinna Friedrich up here, and she is leading the work group on meiotic arrest and male infertility. And while TEX11 was the first gene I ever described 10 years ago, all these other genes have been described in only 10 years by others and us and many in collaborative efforts. And what is shown here is that actually in this phenotype, where in the testis there are no sperm and it only goes to meiosis, we can give a clinically relevant molecular diagnosis in at least 30% of these cases. So this is a high diagnostic rate. This has prognostic value before doing the testicular biopsy, because essentially in all of these genes, the result, the success rate for testicular biopsy will be very low or actually zero. And also it is important for the family, because many of these genes, if they are mutated in women, they lead to premature ovarian insufficiency or POI. So we had the situation now several times actually that we diagnosed a man mutations in such a gene, and if he has a younger sister, we can actually offer to test her, and if she's also at risk to develop premature ovarian insufficiency, maybe we can freeze her eggs and then help her family planning at least. Oh, and I forgot to say, most of these are all autosomal recessive diseases, and some are X-chromosomal. So then another example, and that is now many people are working on this after the first pi RNA. This is a specific pathway working in the testis and keeping these retrotransposons, these jumping genes at bay. And the first gene of this is PNLDC1, and that was published in the New England Journal of Medicine only 2.5 years ago. And since then, we and others are working on this too. And we actually, Birgit Stallmeier, who now heads a whole working group on the pi RNA pathway, she looked into our cohort of, at that time, over 2,100 infertile men, and looked for variants, pathogenic variants in genes associated with the pathway. And to our surprise, and very interestingly, this is nuclear division and meiotic cell cycle. These are the classic meiosis genes, but actually the pi RNA pathway seems almost as important as the things we knew before. And actually through this study, Birgit and us, we could describe in just one approach 7 novel diagnostic genes. They are named here, but again, the single genes are not so important. The concepts are. Now we immediately put them into the diagnostics, and we analyze these 7 genes on top of those that were known before. And just one last mechanism. So this is Manon Ut and Joris Feldman. Originally, he was also from Nijmegen, then he moved to Newcastle and is now in Edinburgh. And they made us aware through their study that actually de novo variants are also important for infertility. So de novo variant means the infertile man carries, just by unfortunate luck, a mutation in a gene, and the parents do not carry it. And this you can basically only find out by sequencing trios, so father, mother, and child. And that is a bit tricky in infertility. And we have added to that also some information. So for instance, this is one case mutated in DMRT1, a gene that also leads to gonadal dysgenesis. And this is the testicular biopsy of these men. And you will, if you are familiar with a testicular biopsy, you will appreciate that this doesn't really look like testis tissue, but more like maybe gonadal dysgenesis. And these are then dominant mutations that can also be so one copy of the gene is impaired, and that already leads to the phenotype. And we have described some others in collaboration with other groups. So just to come to a close and to summarize this, so this was the status in our hands until 2017. So some men have azoospermic men, some have Klinefelter syndrome, some have AZF deletions. We added to that, by analyzing only 20 genes at that time point, 6% relevant diagnoses. And actually that means in 2% you can say, okay, this is obstructive azoospermia, will find sperm in the testicular biopsy, while in almost all others, the success rate of a testicular biopsy is very low to 0. And in reality, now only 2 years further down the road, Corinna, Birgit, and many others here and around the globe, we already have many more genes on our agenda that we have also then compiled lately in a review last year. So I've been talking mostly about these genes, and before there were only 20 and now there are even 40. And this is only 2 years further. So this, I think, clearly illustrates how steep the learning curve is and how fast we are progressing. And just to mention that, so I've talked about CATSPER2. So if a man is mutated in this gene or the phospholipase theta, then he has normal semen analysis, but it requires ICSI to conceive. I didn't talk about the androgen receptor, but just to make you aware, if a man is mutated in that, he might require testosterone substitution. M1AP I mentioned, so the success rate for testicular biopsy is relatively low. And there might be a risk for offspring. We don't know yet. And just finally, these two genes were just published in connection in association with colorectal cancer risk. So a man mutated in these genes, he will not have sperm and amniotic arrest, but he has also cancer risk. And I think this is a topic we need to devote our attention to now and for the future. So if you ask me, it is time to analyze more than the karyotype and the AZF. And to close this, so the summary, my talk is we now have, in contrast to 7 years ago, we have validated infertility gene panels that can inform about the success rate of testicular biopsy. We have this biochemical test that is available, and we have actually doubled the genetic diagnosis in only 7 years. I consider this quite a good achievement. So the final slide is then exome genome sequencing significantly increases the diagnostic yield in infertile men. The genetic diagnosis can predict success rate of testicular biopsy and also medically assisted reproduction. And the molecular pathomechanism, and this I wanted to show you also with the cancer risk, etc., has implications beyond infertility. I mean, we cannot be rest assured yet because this large chunk is still too large, so we need to continue this work. And with this, I would like to thank all the people here in Münster, the local collaborators, and also the International Male Infertility Genomics Consortium, because without this consortium, we wouldn't have seen many of these breakthroughs. And with this, I'm at the end, and thank you for your attention.
Thank you, Frank. I think the questions will be at the end. So please, Virginia.
Yes, as Giovanni says, if you have questions, that was a fascinating talk. Thank you so much, Frank. I'm sure there will be questions for you. If you could type them into the chat, then we can deal with them at the end. But it's now my great pleasure to introduce my old friend, Chris Barrett, who is head of the reproductive medicine group in Dundee, has also qualifications and works as a clinical scientist within the UK National Health Service. Both his bachelor's and his PhD degrees are in zoology, and his early postdoctoral research was at Sheffield University, and it focused on natural cycle IVF. He has been the scientific director of ART centres at Birmingham between the years of 1997 and 2005, and he's been awarded numerous awards for his outstanding contributions to the discipline of andrology, including being made a fellow of the Royal Society of Edinburgh. He was a member of the WHO Male Fertility Semen Analysis Task Force, and between 2012 and 2017 was director of the WHO Male Fertility Expert Working Group, which devised a new system for the diagnosis and treatment of the infertile male. And today, you will be very pleased to listen to his presentation, which is on unanswered questions in male reproduction. Chris, over to you.
Thank you very much, Ginny. It's a pleasure to be here. I'm just making sure that the screen share works okay. Can you see that okay? Does it work? Just checking.
At the moment, we can see your it's like the PowerPoint. It's not a slideshow. It's the PowerPoint with all the controls, but we can see the slides.
Okay. Is that any better?
Well, we can see all the control panels, the task panels.
Oh, right. Okay. I don't know how to get rid of those.
Don't worry, we can see the slide.
Okay. All right. So I'd like to thank the organisers for inviting me to give the talk today. I guess there are many unanswered questions in male reproductive health, so I'm just going to pick a few of those. I don't have any conflict of interest, apart from saying that I'm on the board of two journals, Science and Human Reproduction. So obviously I'd be encouraging people in the audience to look at these two journal formats for publishing their work. So that's my only significant conflict of interest. So as I said, there are many questions facing andrology. And I think the Male Reproductive Health Consortium identified 13 pressing questions facing male reproductive health. And I'm actually just going to pick 4 of those and very quickly go through what I think those 4 key questions are and how we may get answers to those key questions. So the 4 key questions I'm going to pick on are going to be how we diagnose male infertility, and Frank's intimated at that and the sophistication of that. I'm going to talk a little bit about how we develop sperm function testing, which is the second point. I'm going to briefly mention how we may get the new generation of non-hormonal contraceptives. And I think there's fantastic research in that arena at the moment. And lastly, I'm going to say that actually, and Frank showed the example of that from the Male Genetics Consortium, is that the only real way forward we're going to get is actually if we collaborate globally. And I think hopefully I'll be able to make a plea for us to actually do that. So first things first, let's sort of just be very basic in where we are and the challenges. So semen analysis has been around now for over 100 years, actually. And we still don't do it very well. That's the reality of it. And every single external quality control scheme that's published data, and this is examples here from Australia, America, the Netherlands, the UK, etc., and ESHRA, show that actually there's a real problem with just even counting sperm. We don't appear to be able to count sperm particularly well.
Chris, can I interrupt for a second?
Yeah.
You're still on your title slide at the moment. They haven't moved. I don't know if you can move. I'm assuming you must have wanted to be talking.
I've moved them. I've moved. Can you see them moving now?
No, we're still on the title page. I don't know if Brian can help with this.
Okay. It's moving on my text here.
Yes, Chris, if you see the slideshow at the top of this, the toolbar.
Yeah.
It says slideshow, or in the bottom right hand corner, there's it looks like a screen beside the sliding bar.
So none of these are moving when you see them, because I'm moving them now. You can't see anything move. Is that what you're saying? None of the slides move.
Correct. Can you scroll? Oh. Can you move down to the if you click on slideshow tab, can you do that at the top?
Where show taskbar, display settings.
Where it says home, insert, draw, design. If you click on that slideshow.
I can't see where that is. I can't see where that is. I can't see that on my thing. I just says show taskbar, display settings, end slideshow. That's all it says. Right, okay. I can see another one now, but I don't know where it is. That's the problem.
Can you see at the bottom of your screen? There's a sliding bar where it says, I can see it says 60%. There's a line with a plus minus. Can you see that?
No.
Okay.
I can't see that. I'll stop sharing and start again, see if that works. Hang on a second.
Yeah, try that.
Right. Share. Share. Does that come up?
We've still got, try moving it. We've still got your title page and your, yes, it's showing this. Yeah, that's it. It's moving now. But unfortunately, we've got your view as a speaker, so we've got your next slide as well. But at least it's working.
I don't know why that's not work. I don't know why it's not working. I've obviously pressed it.
Chris, if you let me go ahead, I've got your
Yeah,
presentation.
go for it.
I can do it. I'm going to go back to see if this goes.
Meanwhile, while we're organising this, to the participants, mistakenly I said if you've got questions, to type them into the chat. What I meant was you can type them into the Q&A. And I see that some people have already done that, or one person's already done that. There's a Q&A icon at the bottom of the screen, and that's where you should put your questions, not in the chat, because we'll lose them there. We won't notice them. Sorry about that mistake.
Okay, Chris, can you see the screen with your outcome? Okay.
Yeah, I can see that as the main slide now. If you just go through to keep going. That one, stay there. Is that okay? So you're controlling the slides now.
I'm controlling now. Is this where you'd like
to
go?
Perfect. Yeah.
Okay.
So
just let me know when you need to advance.
That's great. So getting back to the main question, which was basically the inability to do a high quality semen analysis. And that's I would use as example one. There is all external quality control schemes throughout the world show that actually, although a sample, which is a pot of semen that's distributed around the different centres, should show similar values, they often show dissimilar values. And the diagram on the right shows the data from the UK NEQUAS scheme, which is about 200 plus laboratories, and that's for sperm count. And what we can see there is that some centres get about 25 million per mil, and other centres get 45 million per mil. And it's supposed to be a similar result. And so clearly there is an issue of doing that. The next slide, please. So standardization, in theory, will help us do this. But of course, standardization has been around for quite some time. The WHO did the original semen analysis manuals in 1980. They have updated that in 2021. And there is an ISO system available now for semen analysis. So that should help us do the different standardization. But there's going to be a question of how we do that analysis and how we do the training. So next slide, please. So there may be in the future, technology might help us. And on this slide, we can see a sperm cell, which we all know stops and starts, moves between different excitement phases. And I've used the examples here of listen to different music. So it's going nice and quietly. It's listening to some classical music. And when it's getting some head banging movement, it's listening to sort of something like Angus Young or whatever. But what we see is different phases, and that's quite difficult to detect by eye. So technology in the future may help us with semen analysis to understand and be able to get a more accurate response. At this moment in time, it can't really do that for semen analysis. But in theory, in the next 5 years, it might be able to do that. So I think example one I would give for the future questions is that semen analysis we don't do very well. We need to do it much better. And in the future, technology might help us actually do that. So next slide, please. So example two is related to not just looking at sperm, but testing their function. And as Frank has shown previously, you get a group of men who've got lots of sperm, but actually they're dysfunctional cells. And the question is, how do we test those cells? On the flip side of the coin, we have men with low numbers of sperm, which are very fertile. And we need to identify those men as opposed to just looking at their sperm count. So next slide, please. So what we see, we've known this information since the 1950s, actually. So this comes from MacLeod and Gould's famous study, looking at fertile and infertile patients. And they show different distributions, statistically different distributions, but there's significant overlap. And the challenge is, how do we identify those sperm cells to look at that particular overlap? How can we detect those differences? The next slide, please. So it would be very helpful for us to do that. And Frank's given an example of the CATSPER assay, where it's looking at sperm motility, and with different formulations, you can actually detect those differences. Be very useful to have that, to have robust, simple, easy to use, clinically cost-effective methods. But at the moment, there are very, very few of those available. The next slide, please. And the example I will give, which is probably the most familiar to you, is looking at reactive oxygen species. So this figure shows a paper from John Aitken nearly almost 50 years ago. And what it's doing is looking at oxidative stress and sperm function. And what it shows with the arrow is when you've got a high level of oxidative stress, you've got poor sperm function. And the diagram on the right, the photograph, which is related to, if you can look at the boots in those pictures, in the 1970s, this is basically how people used to dress going out, and there were no mobile phones, etc. So stepping back in time, this is what we knew about ROS and antioxidant stress. We know there's lots of issues. We knew that reactive oxygen species were negatively affecting fertilization. So we knew that 50 years ago when we used to wear big platform boots. But where have we come since then? So the next slide. So 50 years later, effectively, we've got lots of antioxidants in the market, lots of putative diagnostic assays, but it just doesn't work. So 50 years progress is zero. And John Aitken puts it very succinctly, but he basically says, at this moment in time, diagnostic testing for oxidative stress is rampant uncertainty. And those are his words, not mine. So at this moment in time, the biggest cause of sperm dysfunction, which is basically ROS, we don't know how to detect it, and we don't know how to treat it. So that's an issue. So that's example 2. Next slide, please. Example 3, and I'll briefly mention, where I think there's going to be tremendous progress in the next 5 years, is non-hormonal contraceptives. We've been waiting for hormonal contraceptives for men ever since I was a blastocyst, I think. And they haven't been forthcoming. And there's been, over the last 5 years, a really big push to look at non-hormonal contraceptives in men and targeting the sperm. So next slide, please. So men, and this surprised me, I didn't realize this, but there's only 2 available methods for male contraception, vasectomy and condom. And these have been around for like years and years and years. So there is no developments in this arena, and that's really quite a powerful message that we need to understand. So next slide, please. So in Dundee, and I'll just show 1 or 2 data slides. In Dundee, what we've been doing, funded by the Gates Foundation, is looking at the human sperm and targeting the human sperm with different compounds to see if we can negatively affect sperm function. And the idea there is if we can find those compounds, then we can look at those compounds, see what's of interest, and then we can see how they could affect sperm motility, acrosome reaction. The advantage of doing that is you don't have to shut spermatogenesis off if you're looking at the male, and you can also target the sperm cell in the female reproductive tract. So it has lots of advantages in looking at the sperm cell per se. The next slide, please. So we've developed, or rather in life sciences, they've developed basically an automatic sperm function system where you can basically put the human sperm cell in and you can test, for example, motility, acrosome reaction, hyperactivation, etc., etc. And you can add drugs to those sperm cells and see the response. So you can add drugs that you know what the drugs are supposed to do and see if the sperm cell respond that way, or you can add drugs where you don't know what they do, and then you're going to see a phenotype and what happens to those cells. The next slide, please. So this is the type of readout that we get. This is from about nearly 12,000 compounds. Of course, our interest is contraception. So we're trying to stop the sperm function in the male or in the female. So we have a number of compounds that will stop sperm function, and we can read these compounds out, see what the compounds are, and then hopefully make some progress to what we're trying to do. The next slide, please. So, of course, finding a compound that stops sperm motility doesn't necessarily mean you've got a fantastic contraceptive. Could be toxic, for example. So there's a massively long way to go. However, this approach now allows us to test these compounds, which we never had that before. So with the Gates Foundation and part of the consortium of the Gates global system, then I think that we will have real success over the next sort of 4 or 5 years to getting compounds where they're close to then studying, for example, in first phases in men to see, for example, safety. And I think that's a massive development which we will see. The next slide, please. So the fourth and last example I'll say, which is, I guess, as you get older, you get a bit, in theory, you get a bit wiser, I suppose, is that it's a real realization, I think, that we've been trying to solve problems in male reproductive health for a long time. And we haven't really made a massive amount of progress in some areas. And I think that's because perhaps we haven't maybe collaborated as much together. We haven't approached these in a global method and a global analysis. And the example I will give here is for COVID. So when we had the global pandemic, it's really important to remember that effectively we identified the cause on the 21st of December 2019. And within 12 months, Pfizer had a vaccine which was 95% effective. So in that 12-month period, that scientific and medical progress was absolutely astonishing. But that's because people collaborated. They had a strict focus. There was funding to follow that, et cetera, et cetera, et cetera. So that's the way I think we need to think in the future to solve our problems. The next slide, please. So that's why we've formulated a male reproductive health initiative, a global collaboration, and you can see Frank's picture on that slide as well. We've published a number of papers, strategic papers, saying where we think we are, where we should be going, et cetera, et cetera. And I think that's going to help us identify and push forward on some of these collaborative issues. The next slide, please. However, life always comes down to dollars. And most people you talk to about research funding, particularly at the moment in some countries, research funding is a significant issue. So one of the things that we've looked at is the funding for male reproductive health. So the next slide, please. And what we see here is that funding for male reproductive health, federal funding, either in the United States, Germany, Australia, is very low. So basically it comes to less than 1% of government funding. And this is from 2 papers, one by Yves Gamero and one by Joris and our group for male reproductive health. So it's less than 1% of federal funding is for male reproductive health. And people have always said, well, there's more funding for female reproductive health. Actually, statistically, that isn't actually true. So if you look at the graph on the right, the average award is pretty much the same between male and female. What is very powerful message is the funding for reproductive health is really low compared to the problems that we face. So funding for male reproductive health is low, but funding for reproductive health is low. And that's where a significant problem occurs. And we're only when we start collaborating together and getting a big consortium sorted and answering our questions, will we then make substantial progress. And the funders, bluntly, want to see that type of collaboration in action because they've seen the power of that during COVID. The next slide, please. So I would just summarize, hopefully, there were 13 questions identified by the consortia, but actually I've just given 4 examples, hopefully just to whet your appetite, related to the basic diagnosis, sperm function testing, generation of new contraceptives, attacking the sperm cell, and global collaborations. Thank you very much.
Thank you, Chris. Thank you, Chris.
Yes.
What a powerful message your talk has given us all and lots to think about. So we're now open for questions. I'm going to look and see what we have here. There are 2 questions in the box. And I think this first one is for you, Frank. Whole genome sequence. Can you suggest azoospermia? I'm not quite sure what that question means.
I have seen it and I've been wondering too. I guess the question is whether I would suggest genome sequencing in azoospermic men.
Yeah, that's what I imagine.
Well, not from a diagnostic point of view just yet, I would say. I mean, we are basically doing it by a research project. But at the moment, you do not gain much in comparison to just sequencing the exome, and the exome is still about 1/3 of the costs. So because we just don't understand the non-coding genome very well. On the other hand, I would say if you have a research translational diagnostic setting, then yes, please do sequence genomes in azoospermic men, and let's collaborate and put all this data together, because we are just lagging so many years behind other fields of medicine, where many genomes of many diseases have been sequenced, and yeah, we need to get there.
So I'd just like to add to that. At the very end of your talk, you mentioned the association between a possibility of association between genes for bowel cancer. And is this something that concerns you greatly, concerns me, that if you do extensive genome sequencing, you may hit on things that you didn't anticipate, but that have potentially quite serious consequences. And you need to prepare for that, and how you're going to approach it with the patients where you identify these problems.
Yeah, thank you, Virginia, for coming back to this point. And yes, it both concerns me, but also I think yeah, it intrigues me how to get there. So I think these are actually two points. One is, if we do large-scale sequencing, of course, we have the possibility of findings that are not within the scope of what we're actually looking for. For instance, a typical example is a BRCA mutation, BRCA1 or 2, and that confers a very high risk in women for breast and ovarian cancer. And this, I mean, the genetics community has thought about it for many years, and I understand, I mean, in countries it's handled very differently. For instance, in the US, actionable genes, as they are called, you should actually basically, whenever you have the data, you could look at them. I think in Europe, the Netherlands are very at the forefront of also doing this. In Germany, we are very skeptic on this, and we only do it if the patients explicitly say they want us to look. This is the one side of this topic. The other thing, and that I wanted to make clear, is that the more and more we learn about the genetic causes of male and also, let me emphasize, female infertility, it's very essentially the same. We learn that many of the genes are not just relevant in the testis or the ovary, but actually confer also cancer risks and maybe other disease risks that we don't know yet. And here I think we have a very important opportunity to not only help them and explain why they're infertile, but then also provide tailored screening programs or something like that. But I absolutely agree with you. We need to, and this takes time, explain it to them before we do this large-scale sequencing.
Thank you. We're getting lots of congratulatory messages through in the chat saying thank you both for your excellent presentations. They really have been very, very, very interesting indeed. Certainly for me, sperm is always an area I'm afraid I'm lacking in knowledge. I prefer embryos, and so I'm drawn more to embryos. So when sperm talks come along that I'm forced to listen to, I realize how interesting it all is. So we have another question here which says, "Good day. Thanks a lot for the comprehensive presentations. The first question is addressed to the necessity of conducting the MAR/HbA test. Do we still need these tests?" That's over to you, Chris, I think.
Okay, so I think if I've understand that, MAR, is that sort of antibody testing?
I would imagine so, mixed antibody reaction, yeah.
And HbA, I guess, is the HA. I think that the questions that we have to ask is, are these tests and the way they're presented very accurate, easy to use, cheap, diagnostic sensitive and specific? And the answer probably is no. I mean, we know that for the MAR test, and we know that for the HAB Select, for example, if we look at the paper from that was published in the Lancet. So I think the challenge of these assays particularly, but of other assays, is that you've got to make a sperm function test simple, cheap, but it's got to be effective. And I think the example that Frank gave, which was on Catsper, is that test, my understanding is that it's looking at sperm motility, which everybody looks at every day down a microscope, with two different conditions. They either swim or they don't. Simple. That's what it needs to be. It needs to be simple and effective. And that represents genetic deletions, for example, in Catsper. Those patients don't succeed at IUI and IVF, therefore you need ICSI. So that approach strikes me as the best approach that we can do. The difficulty is that Catsper was discovered in 1999. And actually, it's 20 years research as put together to try and understand that from the genetics and the physiology point of view. I think what we try and do at the moment, and that's just my bias, is somebody has an idea, they have a favorite protein, and they say, okay, let's follow this one because I think it's a good idea, and it doesn't work. You need to follow logically the science. And I think John Aitken's data shows that very clearly. There's clearly an issue with oxidative stress and sperm function, no doubt. It's how do you best measure it, which isn't simple. And secondly, how can you develop a very accurate assay for looking at that that's cost effective, etc. Again, not simple. And we've always tried to make a shortcut. The example I would use, can you just go to the gym once a week and look like an Adonis? Probably not, You've got to do a lot more work. It's not instant. And that's the problem. We have to put the work into it. And that's why you need consortium. And that's why you need a lot of research behind it. And the Catsper test, I think, is a good example of that, of a high quality research. Then you've got a good diagnostic assay. Very long answer. Apologies.
No, excellent. Thank you very much. I mean, goodness me, wouldn't it be wonderful if everybody followed the lead of the Pfizer development of the vaccine and recognized it? Yeah. Classic
example, isn't it?
Completely.
Classic. There's often a scientist and clinicians, you know exactly what we need. You know that. It's not analysing and thinking of the question is not the problem. It's how do we best solve it? And often you need a massive amount of money and huge collaborations. But you can do it. It's not intellectually difficult. And that's what we need. That's just my opinion.
Virginia, can I ask a question to Frank? So, Frank, thank you for the fantastic presentation. Considering clinical point of view, in the vast majority of the cases, we are still at the time of Y microdeletion and karyotype. When do you think that it could be possible to introduce new testing in a clinical setting? What is your idea?
Yeah, Giovanni, first of all, nice to see you and thank you for this. It's depending on how I think about it, it's a tricky question or a very easy question. The very easy side of things is now. I mean, we do it. Some other centers have adopted it too, but I'm well aware that this heavily depends on the basically on the country and the setup. So, for instance, I have always envied Italy because you can prescribe FSH for infertile men. We in Germany cannot. But we in Germany, we can do genetic testing if we have enough evidence. And for these genes that I presented, the evidence is sufficient. So actually, the insurance companies will pay for this genetic test. That makes it, of course, a lot easier. From the researcher point of view and the, let's say, andrologist point of view and independent of payment, I would still argue that we are there to do it. And it always reminds me of, I mean, I have not been in the field at that time point. I was still studying when the AZF deletions were described in the 1990s. And then, okay, very quickly, everyone was convinced that that is an interesting test. But the test that was made up also in collaboration with Münster is a very easy to do test, and it's also very cheap. So that very much, I guess, helped to get it into the clinic. Yeah, and so this was the more complicated answer. My feeling is that maybe I presented now 6% additional diagnosis. In reality, now 2 years down the road, I forgot to mention that actually we now just crossed the 10% additional diagnostic yield, yeah, in azoospermic men. For me, this was one milestone I was waiting for, because then I think we can argue, okay, in every 10th man we can provide a diagnosis. But I mean, Chris is very good in doing these things. I mean, I think we actually have to do a cost-benefit analysis. Yeah. So really calculating a testicular biopsy, also some risks, very low, I know. But yeah, I think these things we still have to do.
So essentially, your conclusion is that you are positive for the future.
Yes, definitely. If people need more convincing, then only a very short amount of time, I would say. Thank you.
Thank you. I think there's another question, maybe for you, Chris. What about the use of antioxidant?
So I think hopefully I showed the difficulties of the antioxidant story. And John sort of isolates it, probably published more papers than anybody else on the planet, I think. It's the challenge is ROS and oxidative damage. Sounds very simple, but it's actually quite a highly complex biology, actually. So identifying what are the key pathologies is one thing. Secondly, making that test widely available is another thing. And thirdly, determining that it's an antioxidant balance is not, again, simple on how you would then get around that. So I think it's a logical thing. You've got oxidative damage, give antioxidants. That sounds logical. You know, A equals B, 1 and 1 equals 2. But actually, it's more complicated than that. And I think what we've done is we've jumped too quickly. We haven't got the diagnosis right. And then suddenly everybody gets antioxidants. And if you look at the survey that the Male Reproductive Health Initiative did of 1,000 patients in which Chris de Jong published, was the main author, what we saw there was that I think there were 56% of men who were on antioxidants. And clearly, they can have a negative effect because they can have a pro-oxidant effect. You can have too much. So I think just giving men with low sperm counts or what's perceived to be oxidative stress antioxidants, every bit of data we have show that just doesn't work. You know, we need to be a bit more sophisticated than that. And I think that's the question. We need to become a bit more sophisticated in how we do that. Because carrying on as we are, it just doesn't work. You can walk around the ESHRE stands. I took a photograph and got in a bit of trouble, actually, of the antioxidants thing. And there was a male model, obviously beat the gym more than once a week. But basically, it was like, you know, eat antioxidants, look like me, your sperm will swim through concrete. And actually, that's not true. You know, it's just not true.
There was a question that's disappeared from my screen, which was talking about fertilovit and various other things. Would they be antioxidants? Is that what these are?
I understand all these, and you know, I don't have shares in anything like that. But I think at this moment in time, the challenge is identifying the patient and providing appropriate, rational treatment. And at this moment in time, you know, John Aiken says it very well. There is rampant uncertainty. And while we've got that, you know, I don't know how to identify a patient any better than anybody else. So actually, it's probably not going to work well. It might work on some people, but it might have a negative effect on other people because of the nature of antioxidants. So I think that's the question that we're faced with. But because it's relatively easy to do, or take some antioxidants, we sort of do it, you know. We need to, you know, go the hard yards. And I think we can only do that when we collaborate well together and we get lots of money for research, and we can then really make fast progress. So we'll be able to identify. And if for tomorrow, if I suddenly was given $30 million to distribute around the world to look at oxidative stress in sperm, I'll bet you in 2 years' time, we'd have a really good answer. And, you know, people like John, Christian, etc., will be able to answer the question. They just need a lot of money and high, powerful research. So we need to work the hard yards to get the answer. Taking the easy route, thinking we can measure something and giving antioxidants isn't working. All the data shows it isn't working.
So to go to Frank again, we have a question. I had used the stem cell therapy for my patients with azoospermia, but I didn't get very good results. Why?
Yeah, first of all, I'm wondering what really the stem cell therapy.
Well, exactly. I don't
is mentioned.
I have to ask that question. It's anonymous attendee, so I don't know. Maybe they can add a little bit to that. Meanwhile, while we're waiting to see if anything happens, we're going to be running out of time. So very quickly, Chris, Can you give us the answer to why is it that people doing semen analyses vary so much?
Well, I think
So many differences in the EQA results.
Yeah, I think the simple answer at this moment in time is training.
Yeah,
I think
exactly.
that's a simple answer. It's just training to do it, and that's relatively easy to do. I think in 20 years' time or 10 years' time, we may have better technology. But at this moment in time, the one thing you can do to improve your diagnosis of male infertility is learn to do semen analysis well.
And do IQAs all the time with your staff?
Yeah.
Yeah.
Basically, yeah, have a comprehensive quality control system. And I think the ISO will help in doing that, for example.
So we don't seem to have anybody adding in what they meant by the stem cell therapy, because that's intriguing. I wonder what they could possibly have done. Frank, can you shed any light on it from your imagination?
I mean, things that I and others are imagining what we could do in the future is, of course, if we understand the genetic cause for, let's say, a defect in meiosis, and as Chris mentioned, the mRNA technology, and a protein is missing there, and because of that missing protein, meiosis stops. Maybe we can introduce this protein into the testis by some mRNA therapy, for instance, and then maybe spermatogenesis would proceed. And I am aware that at least one group is trying that at the moment in primates. And I'm getting a bit, very curious about what we will see. Or in alternative, stem cell therapy could be we take out the stem cells from the testis, we correct them genetically, and put them back. I personally would be a bit anxious and skeptic whether we should actually do that, because it would mean tinkering genetically with our germline. While in the other approach, we would just add some proteins that are lacking. So I would feel more comfortable with that approach.
Well, it looks to me as though we've come to the end of our time. So if we could move on to the next slides, I'd like to thank both our speakers and encourage everybody who's still here in the participating room to invest in your education and join the IFSS community today. We have a QR code here. Share this amongst your friends, especially if you found it useful, which, judging from all the complimentary comments in the chat, I think you all have. I certainly have found it fascinating. Next slide. And I'll pass over to my co-chair, Giovanni now.
Yeah, I think this is the IFFS Congress in Japan. There will be a combined session between ISA and IFFS. Next slide. So this is the Howard Congress that will be held in March from the 29th to the 1st of April. And again, there will be a combined session between IFFS and ISA. So it's a really, really pleasure for us working with you. And I think the successful of this webinar series can justify our collaboration. Thank you for the amazing speaker, the fantastic webinar.
Thank you very much, both of you, and
thank you very much.
Bye,
thanks, bye, Giovanni. Bye.
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 Bye, Frank. Bye, Chris.
Bye.
Bye, Virginia. Bye-bye. Bye, Frank.